Hi, welcome back everyone. This is a very important series of mini lectures because what I will do in these mini lectures is to introduce the vocabulary that you will need to know for the next two chapters that we will cover, chapters 33 and 34. So it's extremely important that you take good notes on this unit and actually work through the study guide 32, before you go on to Module 1 of the Animal Kingdom. So here we go, back to the animal kingdom. Well, one thing that you should remember from the last presentation, that animals have these unique stages of embryonic development. So you, like any other individual animal, we began our individual life as an individual organism, I say the word individual a lot, as a one-celled zygote. All right, again, the zygote is usually formed by fertilization event by two gametes. The male and the female fuse to form the zygote. Then the zygote starts a process of rapid mitosis without cell growth. And do you remember what this is called? Well, it's called cleavage division. So this one-celled zygote divides in half and becomes a two-celled primitive embryo, then the four-celled, then the eight-celled, then the solid ball stage, which is called the morula, and then the hollow ball stage, which is called the blastula. So again, if we look at the 8-cell stage, the whole embryo is no bigger than the original zygote. If we look at the blastula, the whole embryo is no bigger than the original zygote, because even though you have cells dividing, no cell growth. And just to remind you, whenever you have mitosis, mitotic division produces two daughter cells that are genetically identical to each other into the parent cell. And remember, we call genetically identical cells or organisms clones. So in this 8-celled embryo, all these 8 cells are clones of each other. Now, we'll talk about the human example for a little bit, even though, again, this could be an embryo of like a sea urchin or something. If this is a human embryo, let's say we just remove this cell right here. Would we have a kid that's born without an arm? No, actually you could. From the 8-cell stage, you can remove any one of these cells and still have a perfectly normal child. And that is because every cell, and again, this applies to humans, but not all animals, every cell during cleavage division is what we call totipotent. Every cell has its full genetic potential. So in the case of a human animal, every cell here could become heart or brain or liver or any part of the human body, or each cell could become a whole human organism in its own right. So totipotent. And again, if this is a human embryo, then in this nucleus there would be 46 chromosomes, 23 matching pairs, and on each chromosome there are thousands of genes. So in every single nucleus there is the human's entire genome, all 23 matching pairs of chromosomes, all 30,000 pairs of matching genes. But none of these genes have been completely turned off yet, so that's why we can remove a cell and these other cells can just divide and fill in. And sometimes in nature, an embryo will actually divide in half all by itself. And what happens then? If an embryo divides in half, what do you think happens? Do you think you get two half babies being born? No, absolutely not. This is how you get identical twins, because again, these cells are totipotent and they can become any part of the human body in their own right, or a whole human organism. Yeah, that's my terrible drawing. All right, so the blastula is the last stage of cleavage division, and as we'll review, and I think it's module 5C, that a human blastula is called a blastocyst, but that's not important for right now. This is what's important. Cele in biology, C-O-E-L, always means cavity. So this is aptly named. The cavity inside a blastula is called a blastocele. In fact, blastula is just referred to as the hollow ball stage of the embryo. Now, all animals then after the blastula stage form a gastrula. And what happens, so again, this is the blastocele, a second cavity begins to form. I went over this in the last video. It's like if this was a ball of Play-Doh, it's like just like somebody's pushing in their finger right here. Now, the second cavity, if I named it, I would call it the gastrocele, but no. That's not what it's called. All right. The opening right here is called the blastopore. And the cavity that's actually being formed right here, you'll see this on another slide, is called the archeteron. So, unfortunately, there are some vocabulary words in here. Now, you'll notice as this new cavity, the one I want to call the gastrocele, gets bigger and bigger and bigger, the original cavity here, the blastocele, is getting smaller and smaller. All right, as I mentioned before, the gastrula starts to layer. So, any cells remaining on the outside are called ectoderm, and they are always represented as blue cells. And any cells that form this inner layer that line the archeteron, they are endoderm. So let's review these. So the ectoderm, again, is on the cells that remain after gastrulation. These are the cells that remain on the outside of the embryo. And this, again, is virtually happens in all animals' development. And they will form the outer covering of the animal. And again, as this says in human animals, it will form the nervous system. And I realized I forgot to review the key points on on your lecture guide. So, defining characteristics of the animal kingdom. Hopefully you remember we're multicellular, we're eukaryotes, and we get our nutrition by ingestion. We eat, therefore we are animals. Unique protein in the animal kingdom, that's keratin. It's also in your study guide. Unique cells, muscles and nerves. And again, from the last video, I covered this. Life cycle characteristics, our diploid stage is dominant. We reproduce almost exclusively by sexual reproduction. And right now, I'm reviewing and developing a little further the characteristic stages of embryonic development. All right. So, ectoderm. Endoderm, as I already indicated, is the layer of cells that is always designated by yellow. And these will form the animal's digestive tract. All right, so again, this makes sense. If the yellow is going to be the animal's digestive tract, that would mean the opening to the digestive tract here, the blastopore, that is either going to be the animal's mouth or the animal's anus. All right, and finally, some animals stop at this level. They just form an ectoderm and an endoderm. But with most animals, though, some of these cells migrate in and form a middle layer here, which is always represented in red. And this is called the mesoderm. So if you think about it, what's on the outside of your body? Skin and hair. Well, ectoderm is going to form skin and hair. What's the tube going through your body? That's your digestive tract. And what's in between your digestive tract and your skin and hair? Well, muscle, bones, blood, that is what the mesoderm forms. So again, I think I went over these important terms here on your lecture guide. Zygote, morula is the solid ball stage, blastula is the hollow ball stage. As we'll see later, a human blastula is called a blastocyst. We went over cleavage division. We went over the cavity inside the blastula. It's called the blastocele. And we went over gastrula. All right, and blastopore. And archeteron is on your lecture guide, and that is that opening here. We will see that spelled out eventually, I promised. All right, so what we need to go through, also back on your lecture guide, it says back to humanity in the gene idea. The important thing with animals is the Hox genes, which will be talked a little bit more in lecture and in your study guides. But here we get down into the meat of the vocabulary, if you will. So we always classify animals according to their what's called a body plan. And the first way we classify them is what kind of symmetry does the animal have. All right, so there's two main types of symmetry. Well, there's two types of symmetry. There's radial symmetry and there's bilateral symmetry. So a lot of you have a good understanding of that, but let's just clarify. So if an animal has radial symmetry, all right, an example of an animal with radial symmetry is a starfish. Now, we're not supposed to call these starfish anymore. We're supposed to call them sea stars. All right, so if you take a starfish and you put it on top of a pie, all right, do you like pie? My husband just loves pie. All right, now if I put a starfish on top of a pie, I could theoretically cut the pie into wedges and there would be identical pieces of starfish in each wedge of the pie. So that is a good way to picture radial symmetry. Could you put this animal on a pie and slice up the pie that anybody would get the same piece? And then there's bilateral symmetry, in which the body can be cut into left and right halves that are mirror images of each other. So what I'm going to do is show you a little animation that illustrates radial and bilateral symmetry. I'll see you there.